Ever wondered what causes Down syndrome? It's a question that has puzzled scientists for decades. Down syndrome, often known as DS, is marked by an extra copy of chromosome 21. This extra genetic material is associated with a range of cognitive impairments and physical abnormalities. It's like having an uninvited guest at a finely balanced dinner party. It disrupts the harmony and creates chaos. But thanks to the advancements in genomic technologies and molecular biology, we're beginning to understand this chaos. Scientists are now able to delve deeper into the molecular underpinnings of Down syndrome. They're shedding light on the complex interplay of genetic and cellular factors that drive the disorder. The code is slowly but surely being cracked, so it all starts with an extra copy of a single chromosome. But how does this translate to the symptoms we see? Well, let's dive deeper into the molecular mechanisms of Down syndrome. Down syndrome, at its core, is a genetic disorder. It's primarily caused by a condition known as trisomy 21. Now, this might sound complex, but it simply refers to the presence of an extra copy of chromosome 21 in every cell of an individual's body. And this event is typically caused by an error in cell division called non-disjunction during meiosis, which is a process of cell division that produces reproductive cells in sexually reproducing organisms. So what does this extra chromosome do? Well, it disrupts normal cellular processes, leading to a dysregulation of gene expression patterns. Think of it like a finely tuned orchestra. Each musician or gene has a specific part to play. But imagine if one musician suddenly starts playing twice as loud or twice as often. The harmony is lost, and the music, or in our case, the cellular processes, become disordered. Now, researchers have identified specific genes on chromosome 21, such as DIRC1A, DSCR1, and APP, that are overexpressed in Down syndrome. This overexpression contributes to the characteristic features of the disorder. These genes are like the musicians who are playing too loudly, disrupting the cellular orchestra. But it doesn't stop there. Dysregulated signaling pathways, including the NFAT and ERK-MAPEC pathways, also contribute to the molecular abnormalities associated with Down syndrome. These pathways act like the conductors of our cellular orchestra, guiding the activity of our genes. When these pathways are disrupted, it can have wide-ranging impacts, affecting neuronal development, synaptic plasticity, and cellular homeostasis. Now that we understand the molecular mechanisms, let's look at how these translate into the clinical features of Down syndrome. Down syndrome presents a spectrum of clinical manifestations. Central to these are intellectual disabilities and developmental delays that affect an individual's cognitive growth and learning abilities. Physical abnormalities are also characteristic, with distinctive facial features being a common identifier. These include a flattened facial profile, upward slanting eyes, and a protruding tongue. But the impact of Down syndrome extends beyond these features. It's often associated with a host of health complications. Congenital heart defects are common, as are gastrointestinal abnormalities. In later stages of life, individuals with Down syndrome face an increased risk of developing Alzheimer's disease. However, it's important to remember that the severity and presentation of these features can differ greatly from one individual to another. Some may exhibit more pronounced symptoms, while others experience milder forms. It's this range of clinical features that underscores the complexity of Down syndrome. These clinical features vary among individuals, and it's the underlying molecular mechanisms that contribute to this diversity. While Down syndrome is a lifelong condition, advances in research offer hope for improved quality of life. Let's delve into the therapeutic strategies that researchers are currently focusing on. These interventions are targeted, aiming to counteract the specific molecular pathways implicated in Down syndrome's pathogenesis. A noteworthy approach involves pharmacological strategies. But the story doesn't end here. The advent of gene editing technologies has opened a new frontier in Down syndrome research. Early intervention programs are another crucial part of the therapeutic arsenal. These programs focus on cognitive stimulation and physical therapy, and they have demonstrated beneficial effects in improving developmental outcomes for individuals with Down syndrome. In the future, we anticipate a further expansion of these therapeutic strategies, driven by ongoing research and technological advancements. The aim of these therapeutic strategies is to improve developmental outcomes and quality of life for individuals with Down syndrome. And as we look ahead, the future is filled with hope as we continue to unravel the complexities of Down syndrome, inching closer to a world where everyone can thrive, regardless of their genetic makeup. Our understanding of Down syndrome has advanced significantly. We've learned that this condition, characterized by an extra copy of chromosome 21, disrupts normal cellular processes and leads to dysregulation of gene expression patterns. Key genes on chromosome 21 such as DYRK1A, DSCR1 and APP have been identified, and their overexpression contributes to the diverse manifestations of Down syndrome. These insights into the molecular underpinnings of Down syndrome have not only deepened our understanding of this disorder, but have also guided the development of targeted therapeutic interventions. These interventions hold the promise of addressing the diverse clinical manifestations of Down syndrome and improving the quality of life for affected individuals. Moreover, the continued focus on unraveling the molecular mechanisms underlying Down syndrome is critical. Research in this field is a beacon of hope, illuminating the path towards the development of novel therapeutic strategies. Unraveling the complexities of Down syndrome continues to be a promising area of research, with the potential to significantly improve lives.